everybody who organized this and for inviting me. I wanted to start with a little bit of history. First, I have to acknowledge some people. Tom Pratt, who's the chair of the Lake Superior Technical Committee, was an unwavering supporter of our efforts to put together the food web analysis and ecosystem model. Don Schreiner, Mark Vincent, Dan Ewell, all helped me beat my head against the wall of writing proposals to not get funded to do this. And then a bunch of other people started the process back in the late 1990s. Jim Kitchell's lab and 3 postdocs started really what I would call the proto-CSMI, where there was a series of cruises I was lucky enough to participate in that really started a cooperative effort to not only accumulate all of the knowledge that we had on fish, but also start to consider zooplankton and even phytoplankton and other, the base of the food web in a lot of our consideration. That led to a series of things. Jason Stockwell, Mike Searson, again, Don Schreiner, a variety of others were involved with the planning for the 2005-06 CSMI sampling. Back then we called it the lower trophic level monitoring. That led us to a bunch of data sort of coming together about 2007 and 2008. Jason Stockwell and I and Dan Ewell sat down, and especially with a data set that was produced by one of my graduate students, Allison Gamble. She had dissected over 10,000 fish to look at the diets. Once we had the diets and all of the other information, we really had a chance to go back and look at the early food web that the Kitchell lab put together and the ecosystem modeling framework called EcoPath with EcoSim. Having been involved with that effort, I knew that the data pedigree was relatively low, and seeing the data that we had in hand, it was much better. And so that was what started all of this. And then Daniel took over the reins, which eventually slowed down. And in about 2015, he sort of called me and said, maybe we should get a postdoc to work on this. So Joel Hoffman and I eventually shared the funding responsibilities for a postdoc named Brian Mathias, who was a brilliant wielder of this software called EcoPath with EcoSim, and put together several iterations of the food web. And just if you are going to attempt to put together a food web, there is a little bit of a Rorschach test that you should go through before you land on the finished version. So if it looks like an elm tree, it's probably not going to balance, which means you're really complicated on top and you forgot about a lot of the details on the bottom. If you put one together that looks like a plate of spaghetti with meatballs in it, or an amoeba, that's not going to balance either. There's too much detail in it. If it looks like a butterfly, on the other hand, it has a chance. By incorporating the complexity in the lower food web, the model actually balanced. Now, having been involved with Sean Cox's version of the original one that the Kitchell lab put together, there were a lot of different things that he had to really lever in place and fix to get that to balance. This one actually balanced pretty easily. So we're confident that we've captured the complexity that we needed, and there's some details that we had to leave out because they were essentially causing chaos. So there's a parsimony here that I think fits. Just to explain a little bit about this, there's several different ways to show this. In this case, the colors match up with the category of organism. These are actually detritus sources, and we split this into nearshore and offshore. Again, there's a magical delineation at 80 meters. I think it's pretty close to really what happens in the lake. At the center of the universe are Cisco, of course, which tend to be using energy from both nearshore and offshore, as Tom mentioned. And then there's another series of, you can see some really small dots here. These are the charismatic megafauna of the lake, which are the Pacific salmonids or or steelhead and so forth. We kept them in the model because as climate continues to change, they may become important later, but right now they're meaningless. I like to catch them, so they have some meaning, but not in the model. The really important aspects of this are the microbial loop. So here we have ciliates, nanoflagellates, and bacteria. Bacteria. These are the connections, if you remember from limnology, the connections to Ray Lindemann's ooze-centric food web trophic dynamic concept. And those connections to the detritus are really the only way you can explain the energy in the system. Those of you who study microbial organisms, I've seen the light. The fish and zooplankton guy now knows how important you all are to the universe. And so without this, there's really no way to explain how we get the rest of the system, at least in a meaningful way. Using this framework is the only way to actually assess properties of the ecosystem, and we're working on transfer efficiencies, super efficient transfer. And also constructing the food web as we did, we're addressing climate change issues by looking at consumption of biomass.